Welcome to part 19 of this video on the immune system. Okay, so in this video what we're going to begin looking at is B lymphocytes, and I want to discuss the same things for B lymphocytes as we did for T lymphocytes. Specifically, I want to look at the production of B lymphocytes because I think this is necessary for a complete understanding of how the adaptive immune system works. Okay, so the production then of B lymphocytes. So the start of the story is the same as for the production of T lymphocytes. So again, we're going to start our story in the bone marrow. So I'm going to redraw this picture out here. So here is one of the long bones which will contain some bone marrow, and of course I've chosen the femur to draw here because it's a nice, easily recognizable bone. Okay, so here's the femur, and then in the middle of the femur you'll have a space where the bone marrow is present. So around the edge this will be the compact bone, and then in the middle of the shaft you've got an area where bone marrow is present. So I'll just colour this in here in red. And of course, one of the major cell types that is within the bone marrow is going to be the hematopoietic stem cell, and we've already seen that the hematopoietic stem cell is responsible for producing lymphoid progenitor cells. So I remind you that the hematopoietic stem cell will divide and divide and divide, and some of its progeny will remain as hematopoietic stem cells to maintain the population of hematopoietic stem cells within the bone marrow, whilst others will then differentiate into new cell types. Okay, so within the bone marrow we will have these hematopoietic stem cells, and these are going to give rise to lymphoid progenitor cells, and it's the lymphoid progenitor cells that not only produce us T lymphocytes but also produce us B lymphocytes. So these are the precursors to both T and B lymphocytes. So we're going to get lymphoid progenitor cells, and now here's where the story is going to go differently. So remember, in the production of T lymphocytes, lymphoid progenitor cells then left the bone marrow, went into the bloodstream, and then went up to the thymus gland. In the production of B lymphocytes, it's different. The lymphoid progenitor cells are going to remain in the bone marrow, and this is key because this is the reason that the production of B lymphocytes can actually continue throughout your entire life, because it's not reliant at all on the thymus gland, so it's not affected by thymus gland involution. So the lymphoid progenitor cells are going to remain in the lymphoid progenitor cells that are going to become B cells are going to remain in the bone marrow for their development process. So they are going to stay here, they're not going to leave, unlike the ones that are destined to become T cells. So they're going to stay in the bone marrow. And what are they going to do in the bone marrow? Well, remember what the first step in producing a T cell from a lymphoid progenitor cell was. It was to design the T cell receptor. The first step in becoming a B cell, the major step in becoming a B cell, is that we now need to give each one of our lymphoid progenitor cells the opportunity to design its own B cell receptor. It needs to create a B cell receptor. And B cell receptors for short are abbreviated down to BCRs. And just like T cell receptors were responsible for a T cell being specific to a certain antigen fragment of a specific antigen, the B cell receptor is responsible for making a B cell specific to a certain epitope of an antigen. Remember, there is a big difference. The T cell receptor recognizes a fragment of an antigen, whereas B cell receptors, they are going to recognize the intact antigen, i.e. in its normal folded state, and they're just going to recognize a little portion of that, which, as we discussed right at the beginning, is known as an epitope. Okay, so BCR stands for B cell receptor. And now what I want to discuss in a huge amount of detail is what are these B cell receptors? And this brings us on to the topic of immunoglobulins. Okay, so we're now going to discuss immunoglobulins. So firstly, let me define what is meant by an immunoglobulin. Actually, firstly, let me just straighten up the piece of paper. So we're coming on to this topic of immunoglobulins. So what is an immunoglobulin? Well, an immunoglobulin is a molecule that is associated with B cells, and there are two different forms of immunoglobulins. There is the secreted form, and this is what we would know as antibodies. So when we talk about antibodies, these are secreted immunoglobulin molecules. So secreted immunoglobulins are antibodies, whilst membrane-bound immunoglobulin, so membrane-bound immunoglobulin molecules, these are what we would call B cell receptors. So each of these lymphoid progenitor cells that is going to develop into a B cell is going to design its own immunoglobulin, and it's going to use this to make membrane-bound immunoglobulin, which will be the B cell receptor on its surface. And it can also make secreted immunoglobulins, which will then be antibodies, which are then the actual effectors. They're what, once we've actually activated the B cells and made effector B cells, you will get going into the bloodstream and going to the site of the infection and helping to clear the extracellular pathogen. Okay, so immunoglobulin covers both antibody molecules and B cell receptors, because the B cell receptor is actually a very similar molecule to the antibody molecule. They're both immunoglobulins. There's a very small modification to the immunoglobulin molecule to make it a membrane-bound immunoglobulin molecule over a secreted immunoglobulin molecule. Okay, right. The next thing I want to show you is a simple picture of what an immunoglobulin molecule looks like. Then I want to talk about the different classes of immunoglobulin molecule, and then I want to draw you a more complicated picture of the structure of an immunoglobulin molecule, and I hope that through this discussion we'll gradually get more intuition about these complicated molecules. So, firstly, a simple picture of what an immunoglobulin molecule looks like, and we've seen this picture before in our discussion of the classical complement cascade, but now we're going to discuss it in much more detail. So this classical Y-shaped structure, like so. Okay, and I'm going to put a line there, a line here, and two lines here. So now let me explain this picture. This is the classical picture to draw for an immunoglobulin molecule, and each of the lymphoid progenitor cells is going to have to design an immunoglobulin molecule which it will use to make its B cell receptor and use to make secreted antibodies if it ever becomes activated. So these lines that I'm now colouring in orange, these are what are known as the heavy chains of the immunoglobulin molecule. So overall, an immunoglobulin molecule is made out of 4 separate polypeptides that are bound together, and these lines here are the bonds, and they are disulfide bonds. So there's one disulfide bond between this polypeptide and this polypeptide, 2 disulfide bonds between the 2 polypeptides in orange there, and 1 between this one and this one, and it's perfectly symmetrical down the middle line there. Okay, so these polypeptides that I've coloured in orange here, these are what are known as the immunoglobulin heavy chain. And by the way, the abbreviation for immunoglobulin is Ig, so I could call this the immunoglobulin Ig heavy chain. And the 2 immunoglobulin heavy chains that I've got making in this immunoglobulin molecule are going to be identical. So we will see that each of the lymphoid progenitor cells will have to design its own immunoglobulin heavy chain, and then it will produce loads of those, and it will produce identical ones, of course, and it will attach 2 identical ones together to make an immunoglobulin molecule. But you need more than just 2 immunoglobulin heavy chains to make an immunoglobulin molecule. You also need immunoglobulin light chains, which, as you can see, are the smaller polypeptides. They're in red. So these are what are known as the immunoglobulin light chains, here in red. And again, each of these lymphoid progenitor cells that is going to become a B cell will have to design its own immunoglobulin light chain, and we will go through this design process in a huge amount of detail, and it will come up with a design for an immunoglobulin light chain. It will produce loads of identical copies of this, and then it's putting 2 of those onto the immunoglobulin heavy chain dimer to finish the immunoglobulin molecule. So this is an immunoglobulin molecule. So 2 immunoglobulin heavy chains that are identical, and 2 immunoglobulin light chains that are identical. Now, the key thing about immunoglobulins is that they are capable of binding to other proteins. They have a site that is specifically there to bind to other proteins, and of course what we want is to produce B cells that are going to produce immunoglobulins that can bind to antigens, proteins from pathogens, pathogen antigens. So if I just draw a pathogen's antigen here, so let's have this as our pathogen's antigen. I've now drawn it in the binding site for the antigen. So these portions right at the top, these are known as the antigen binding sites. And of course, because the molecule is completely symmetrical down this midline here, if we've got an antigen binding site up here, we must also have an identical antigen binding site up here. So indeed, immunoglobulin molecules are bivalent. They can bind to 2 antigen molecules. So here in yellow, this is representing the antigen molecule, and I'll just put AG here for antigen. Okay, so A little g like that is just an abbreviation for antigen. Okay, and I remind you that immunoglobulins, their antigen binding site, this portion up here, this will bind to the antigen whilst it's in its native form. So you don't need to chop up the antigen in the way that you did for T-cell receptors. It's not fragments here, it's the native antigen. And the specific little site on the antigen that the antigen binding site of the immunoglobulin molecule binds to, that has the special name epitope. So I'm just reminding you of some of this key terminology here. Okay, right. So, a little bit more terminology about the immunoglobulin heavy chains and the immunoglobulin light chains. So we can divide the immunoglobulin molecule up into 3 different regions. So I'm putting these dashed lines here. So these portions above the dashed lines, this portion here and this portion here, these are the antigen binding sites, or we can call them the variable portions of the immunoglobulin molecule. So this is the variable region, or probably a better description of it is just to call it the antigen binding region. So again, I'm using my abbreviation for antigen there of AG. So this is the antigen binding region. So why have I called it the variable region? Well, think all of the different lymphoid progenitor cells that are going to develop into B cells, they're all going to design their own immunoglobulin heavy chains and their own immunoglobulin light chains. That must mean that there's a portion of the immunoglobulin heavy chain and the immunoglobulin light chain that is going to be different between the different lymphoid progenitor cells once they've finished in this design process. These are the regions of the immunoglobulin heavy chain that are going to be different between the different finished designed portions. Okay, so the variable region of an immunoglobulin molecule is the bit that will be different between the different B cells once you've finished producing them. So they'll all have their own variable region for their immunoglobulin molecule. And that's because they're all going to produce immunoglobulins that will bind to different epitopes on potentially different antigens. Okay. This portion, meanwhile, so this sort of smaller y, if you like, so underneath the 2 lines here, this then is called the constant region of the immunoglobulin molecule, and this isn't going to be different between the different B cells. Once the lymphoid progenitor cells have finished making their heavy chain and their light chain, this portion will be the same, although as we'll discuss, there is a complexity here because of the different classes of immunoglobulin molecules, and that makes discussing this so much more complicated. But for now, just think of that portion as the bit that isn't going to vary between the different finished B cells once we produce them. Okay, so to take this a little bit further, we can refer to this portion of the immunoglobulin heavy chain, this portion here and this portion here, as the variable portion of the immunoglobulin heavy chain. We can then refer to this portion here as the constant portion of the immunoglobulin heavy chain. This is the portion that each of the different developing lymphoid progenitor cells, which are going to become B cells, will design and will come up with a different design for, whereas this bit they're not allowed to touch. This bit is the same for all the different cells. Similarly, with the immunoglobulin light chain, this portion will be the variable portion of the immunoglobulin light chain, and this portion will be the constant portion. Okay, so we have variable and constant portions for both the immunoglobulin heavy chain and the immunoglobulin light chain. The variable portions make overall the variable regions, which are the antigen binding regions of the entire immunoglobulin tetramer, and the constant regions make overall the constant region of the immunoglobulin molecule. Okay, the final thing that I want to mention is that you can produce the two different forms of the immunoglobulin molecule. So let's suppose our lymphoid progenitor cell has come up with a design for its immunoglobulin molecule. It's designed its immunoglobulin heavy chain, it's designed its immunoglobulin light chain. Now we want to understand how can you produce these two different forms of the immunoglobulin molecule, a secretive form and a membrane-bound form. Well, you can make a modification to the immunoglobulin heavy chain. You can stick an additional little bit on the immunoglobulin heavy chain, like so, which will be the membrane-spanning alpha helix, the bit that will implant into the membrane. So you can choose when you're producing an immunoglobulin heavy chain according to your design, whether you want to include the membrane-bound portion, the bit that will span membranes, or not. And this is not a modification that will be made on the level of the DNA. This is a modification that will be made at the level of splicing. So once you've designed your immunoglobulin heavy chain, when you're actually transcribing it, you'll produce a nice big piece of mRNA, and this will all become more apparent later on. And then at the splicing stage, you can either choose to leave in the bit on the constant region of the immunoglobulin heavy chain that would allow the immunoglobulin molecule to implant in a membrane, then you would overall produce a membrane-bound immunoglobulin molecule, or you can splice it out then, and then you produce the secreted form of the immunoglobulin, the antibody. So I hope that's clear that the difference here is not at the level of the DNA. So you don't, each of the lymphoid progenitor cells does not have to decide whether it's going to produce membrane-bound form or secreted form. It maintains the ability to produce both of them forever, okay? And it will produce both of them. It will be able to produce both of them. It will firstly, of course, just produce the membrane-bound form, because before it's been activated as part of the adaptive immune response, it doesn't need to produce the secreted form, it just needs to produce the membrane-bound form to make its B-cell receptors. But once it's been activated, it can then alternatively splice the mRNA and cut that bit out that would produce the membrane-implanted portion of the immunoglobulin heavy chain. Okay, right. So now I'm afraid it gets even more complicated. I've made this a little bit too simple. We're going to now have to talk about the concept of the different classes of immunoglobulin molecules. Okay, so here we go. An additional complexity, and then we'll go on and draw our more complicated picture of what an immunoglobulin molecule actually looks like. So the different immunoglobulin classes. So there are different classes of immunoglobulin molecule. And I'm sure if you've ever studied anything about immunology, you will have heard of these. So there are IgA molecules, immunoglobulin A molecules. There are IgD molecules, immunoglobulin D molecules. There are IgE molecules, immunoglobulin E molecules. There are IgG molecules, immunoglobulin G molecules. And there are IgM molecules, immunoglobulin M molecules. And these are known as the different immunoglobulin classes. And what I want to now explain is what is actually going to change between these different immunoglobulin molecules. So, our lymphoid progenitor cells that are going to develop into B cells and are still in the bone marrow, they will have to design the variable region of their immunoglobulin heavy chain and the variable region of their immunoglobulin light chain. So they will design a specific antigen binding region for immunoglobulin molecules. Each of those lymphoid progenitor cells, once it's done that, and after it's done that, it will be well on its way to being called a B cell. So each of those developing B cells will then still be capable of producing every single class of immunoglobulin molecule, and for every single class of immunoglobulin molecule they'll also still be capable of producing a secretive form of it and also a membrane-bound form of it. Now, what is actually going to be different between the different immunoglobulin classes is the constant portion of the immunoglobulin heavy chain. So once you've designed your variable region of the immunoglobulin heavy chain, you always use that when you're producing an immunoglobulin heavy chain. However, there is a complexity in that there are lots of different choices for which actual gene segment, and we'll see all of this, we'll look at the genetics of this later on, for which gene segment you actually use to make the immunoglobulin heavy chain, and there are lots of different choices. Overall, I think it's 10, let me just count, yes, 10 different immunoglobulin gene segments that you can use for the heavy region of the immunoglobulin heavy chain, and these do produce then different immunoglobulin molecules. So if you use two heavy chains, and I should state that when you're building an immunoglobulin molecule, the two heavy chains that you will use, they will have to be absolutely identical, so their constant regions will also have to be identical. Okay, and for each of these different constant regions that you can use, there will also be the option to have it either with the membrane binding region or not with the membrane binding region. Okay, so don't worry if this isn't completely clear yet. It's a complicated concept. I hope it will become completely clear as the video goes on. So, it gets even more complicated than just having the 5 here, because there are actually, in each of these classes, these are just the headings, there are different subclasses for some of them. So actually for IgA there are actually 2 different types of immunoglobulin molecule that we put under the heading IgA molecules. There are IgA1 immunoglobulin molecules and there are IgA2 immunoglobulin molecules. So these are now specific types of immunoglobulin molecules. This is a heading, and both of these 2 different types of immunoglobulin molecules are fundamentally different, but they're put overall under this heading of immunoglobulin A, and we would refer to these as now subclasses of Ig molecules. Okay, now it's more simple for IgD. IgD, there is just one type of IgD, and that's just called IgD. Okay, and in fact let me add a little bit more information on here. So if you're making an IgA1 molecule, what you do is, let's say we've got our developing B cell and it's designed its immunoglobulin heavy chain variable region, and it's designed its immunoglobulin light chain variable region. What you will do is you'll produce 2 immunoglobulin light chains, okay, and then when you're producing 2 immunoglobulin heavy chains, you'll have to decide, there is a choice of which constant region you're actually going to use. And if you're going to make an IgA1 molecule, you use 2 constant regions that are known as constant alpha 1. Okay, so for both of these immunoglobulin heavy chains, you're going to use the constant alpha 1 gene segment, the C alpha 1, alpha for A here, C alpha 1 gene segment, and that will code for a certain structure for the immunoglobulin heavy chain, and then the immunoglobulin molecule that you overall end up with then will be known as an IgA1 molecule. And for this type of molecule, you can choose on the constant alpha 1 whether you want to have the membrane implanting portion or not. So you can either say, okay, I don't want to have that membrane implanting portion, and then you'll get a secreted IgA1 molecule, or you can say yes, put the membrane bound portion on, and you'll then get a membrane bound form of IgA1, i.e. a B cell receptor IgA1. Okay, so I hope this is becoming clear. I'll go through all the others as well. So for IgA2 the exact same thing holds, but now a different constant gene segment, and I will show you the DNA of how this works later on, but for now just watch the names. So don't worry if you're not fully getting this, you will get it by the time we see the DNA. Okay, so for IgA2 immunoglobulin molecules, you use the constant gene segment known as constant alpha 2 to make the constant region of the immunoglobulin heavy chain. So if we're dealing with the same developing B cell, then the variable region of the immunoglobulin heavy chain will be exactly the same. Okay, but now we're going to use a different constant region for that immunoglobulin heavy chain. We're going to use the constant alpha 2 region, or constant alpha 2 type form of the immunoglobulin heavy chain, and therefore the immunoglobulin molecule that we then end up with is called an IgA2 molecule. And again, for this C alpha 2 type, you can choose to either have the membrane implanted portion or not have the membrane implanted form, and you'll either then get a secreted IgA2 molecule or a B cell receptor form IgA2 molecule. And the principle is exactly the same for IgD. There is a constant portion for the immunoglobulin heavy chain known as the C delta portion. Okay, so if you use two immunoglobulin heavy chains that have as their constant region the C delta region, then we end up with an immunoglobulin molecule known as an IgD molecule. Now going on, IgE, again there are two different types of IgE. There is an IgE1 molecule and there is an IgE2 molecule. For the IgE1 molecule, you have used immunoglobulin heavy chains where the constant region of those immunoglobulin heavy chains is the constant epsilon 1 region. For IgE2 molecules, you have used two immunoglobulin heavy chains where the constant region for those immunoglobulin heavy chains is the constant epsilon 2 region. Okay, so I'll continue on highlighting it up. So we've overall seen 5 different types of immunoglobulin molecules now. Well, 10 if you count the secreted and membrane-bound forms as different. Okay, now IgG, this is the one where there are the most different subclasses. So there are actually 4 different types of IgG. So there's IgG1, there's IgG2, there's IgG3, and there is IgG4. So an IgG1 molecule, that's an immunoglobulin molecule where you have used for the constant region of the immunoglobulin heavy chains the constant gamma 1 portion, or the constant gamma 1 form. And IgG2, it's the constant gamma 2 gene segment that you'll use to actually code for the constant region of your immunoglobulin heavy chains. For IgG3, constant gamma 3. For IgG4, constant gamma 4. Again, I'll just highlight them up. So these are the types of immunoglobulin molecule, and these are the names for the different constant regions that you have actually used in your two immunoglobulin heavy chains that make up the immunoglobulin molecule. Finally, it's a bit of a squash here. There is just one form of IgM, so that's nice and simple, and that will be an immunoglobulin molecule where the immunoglobulin heavy chains, the two immunoglobulin heavy chains that you've used to make it, will have as their constant region a constant mu region. You have used the constant mu gene segment to actually make that immunoglobulin heavy chain constant region. Okay, and all of these, I'll stress again, all of these different types of immunoglobulin molecule that you can build, each of these constant gene segments will have a portion that codes for the bit that would implant the heavy chain into the cell membrane, and that bit can, when we're at the level of the mRNA, can either be spliced out or not spliced out to produce the membrane-bound and secretive form of the immunoglobulin. Okay, so I hope that that is now clear what the different classes of immunoglobulin are. If it's still not clear, don't worry, we will come back to this when we look at the genetics of the immunoglobulin heavy chain, and I hope after you've seen that, then it will be absolutely clear. Okay, so what's the significance of this is what I want to ask now. So we can build these immunoglobulin molecules where we're using loads of different structures for our constant region of the immunoglobulin heavy chain. Does that affect the function of the immunoglobulin molecule that you've produced? Do these different classes of immunoglobulin molecules, and overall we've got 10 different types of immunoglobulin molecule here, do these 10 different types of immunoglobulin molecule have different functions? And the answer is yes, but we won't come to those functions right until the end. So we'll come to the different functions right at the end. For now, what I want to look at is, do they have different structures? And the answer is they do have slightly different structures. So what I'm now going to do is draw a more in-depth picture of an immunoglobulin molecule. And now that we've discussed all these different types of immunoglobulin molecules, we can now see where the structure is actually going to be different for these different types of immunoglobulin molecules. Okay, so what I now want to show is all the different domains of an immunoglobulin molecule, because it turns out that the light chain is made out of 2 important domains, and the heavy chain is made out of either 4 or 5 important domains, known actually as immunoglobulin domains, which will depend, and the number of these immunoglobulin domains that you'll have in the heavy chain will depend on which type of constant region that you've actually used to make that heavy chain. Okay, so these special domains then, these are known as immunoglobulin domains. So the immunoglobulin light chains and the immunoglobulin heavy chains, their structure is made up for the most part of these immunoglobulin domains. These are the really important parts of the structure of these molecules. So, as I say, the light chain will be made out of 2 immunoglobulin domains, and the heavy chain, it will depend on which type of immunoglobulin molecule we're actually dealing with and which constant region, what type of constant region you've actually used for that immunoglobulin heavy chain. Okay, so let me draw this here. So we'll start off by drawing the light chain then. So here is the first immunoglobulin domain of one of the light chains of an immunoglobulin molecule here. So here's the amino terminus, and here's the C terminus. Then what we're going to do is we'll have the immunoglobulin heavy chain next to this. So here is the immunoglobulin heavy chain's N terminus. Then here is another immunoglobulin domain. Then we come to this hinge region of the immunoglobulin heavy chain. So where we suddenly move around, this is known as the hinge region of the immunoglobulin heavy chain. So I'll just actually write that down. So key term, the hinge region is this region here, between the second immunoglobulin domain of the heavy chain and the third immunoglobulin domain of the heavy chain. So that's the hinge region. So here's our third immunoglobulin domain of the heavy chain, and here's our fourth immunoglobulin domain of the heavy chain. And for most types of immunoglobulin molecules, it would then end at this point, but we'll discuss that for IgE and IgM it would go on, but I'll do that in just a moment after I finish the picture here. So now what I'm going to do is draw another immunoglobulin heavy chain, like so. So here's its C terminus, and then we're going upwards. So here's the second immunoglobulin domain, the first immunoglobulin domain, and then the N terminus. Unfortunately, I haven't managed to completely draw this symmetrical, but never mind. And here then is another one of these immunoglobulin light chains. Okay, so there's the N terminus, and there is the C terminus. Okay, so let me now colour this in to make it look a little bit more interesting. So we'll have the immunoglobulin domains of the immunoglobulin light chains in pink here, like so. And there are names for these different immunoglobulin domains of the light and heavy chains. So this immunoglobulin domain, right up at the amino terminus of the light chain, and of course it will be identical to this one. Remember, these two proteins are absolutely identical. This domain is known as the variable domain of the light chain. Okay, so we would call it V subscript L. So the subscript is denoting that we're talking about the light chain. So this is the variable immunoglobulin domain of the light chain, and of course this is the one that all of the different developing B cells can design differently, and we will discuss that process in much more detail later on. Okay, so that's the variable immunoglobulin domain of the light chain. Then this next immunoglobulin domain here, this is the constant immunoglobulin domain of the light chain. So this is the one that won't vary between the different developing B cells. So you do not design this one. This one always remains the same. This is the one where you can play about a bit and come up with your own design. Because these two up here, these two immunoglobulin domains, these make up the antigen binding domain of the entire immunoglobulin molecule. Next, let me now colour in the immunoglobulin domains of the immunoglobulin heavy chain here in green. So as you can see, we've got 4 immunoglobulin domains, usually for an immunoglobulin heavy chain, as I'll discuss some of the immunoglobulin heavy chains, some of the different types. For the constant region of the immunoglobulin heavy chain, we'll have an additional immunoglobulin domain. Okay, so let me give you the names of these different immunoglobulin domains then for the immunoglobulin heavy chain. So this one up here, this is known as the variable heavy immunoglobulin domain. So this is the variable immunoglobulin domain of the heavy chain. So we subscript it H to denote that we're now dealing with the heavy chain. Okay, and again, that's the portion that each one of the developing B cells is going to design and come up with its own design for. So together these two will be unique to each of the developing B cells. They'll all have their own design for it. They'll all produce slightly different domains here, and therefore these will bind together to form a slightly different antigen binding domain. Okay, next down we have the constant heavy chain 1 domain. So this is the constant immunoglobulin domain, the first constant immunoglobulin domain of the heavy chain, denoted CH1. And this will not be allowed to change. So each of the different lymphoid progenitor cells will not come up with their own design for this. However, this will be different depending on which of these 10 gene segments you use to code for the constant portion of the heavy chain. So the first constant immunoglobulin domain of the heavy chain will vary between different immunoglobulin molecules depending on which of these different constant regions you've actually chosen to use. Okay, then it just goes down logically. So this is now known as CH2 domain, and this is now known as the CH3 domain. Now, what do I want to say about the different immunoglobulin classes? Well, most of these different types of constant region of heavy chains that you can use have just 3 immunoglobulin domains. However, some of them, specifically the constant epsilon 1, the constant epsilon 2, and the constant mu types of constant region for the immunoglobulin heavy chain, they actually have 4 immunoglobulin domains. So you'll then have a 4th CH domain. So you'll actually have CH4. So imagine sticking another one on here. That would then be your picture for what an IgE immunoglobulin molecule or an IgM immunoglobulin molecule would look like. But for all the others, so for all the IgA types, for the IgD immunoglobulin molecules, and for all the IgG immunoglobulin molecules, this is the picture that you draw. So when you use one of these gene segments, so constant gamma 1, constant gamma 2, constant gamma 3, constant gamma 4, constant delta, constant alpha 1, or constant alpha 2, to code for the constant region of the immunoglobulin heavy chain, it will produce you a protein with just 3 immunoglobulin domains in, like what I've drawn here. Just to finish this picture up, I'll just put in the disulfide bonds. So we'll have 1 disulfide bond, there, 1 disulfide bond there, and 2 disulfide bonds here. The final thing I want to remind you of is that for each of these 10 different forms of the immunoglobulin molecules that you can produce them in either the secreted form or the membrane-bound form, and all that involves is then producing a splicing level change to the mRNA that you've produced. So each one of these gene segments that can code for the constant region of the immunoglobulin heavy chain, it has a portion in that would code for the bit that would implant the immunoglobulin heavy chain in a cell membrane. And that will be transcribed and it will be part of the mRNA, and you can either cut that out in the splicing process, and then the immunoglobulin heavy chains that you'll produce will not have the membrane bound portion, and therefore the immunoglobulin molecule that you'll produce will be a secreted immunoglobulin, or you can leave it in, and then the immunoglobulin heavy chains that you produce, they will bind in membranes, and the immunoglobulin molecule that you'll produce will be a membrane-bound immunoglobulin molecule, i.e. a B-cell receptor. Okay, so that's my basic introduction to immunoglobulin molecules and all the different types of immunoglobulin molecules. What we'll do in the next video is we will discuss the designing of the immunoglobulin heavy chain and the designing of the immunoglobulin light chain. We'll go over all the genetics, and I hope that will make this concept of the different classes of immunoglobulin molecules more clear.